Hello, everybody. I think we're all set. So let me just say before we get going for real, if at any point the audio cuts out or anything, just let me know. Yeah, so here we are. Welcome back to Evolution for Creationists. As always, I'm just going to listen for a second, make sure everything clicks in, everything's good, and it sounds great. So, let's get started. So we are now up to episode 4 of Evolution for Creationists, which is on Mendelian genetics today. So last time, if you remember, we looked at Mendel's experiments and how he showed that the earlier kind of consensus idea about inheritance was wrong. That was the blending inheritance hypothesis. Today, we're going to say, okay, we know what's wrong. Now let's look at the model that Mendel developed to replace the blending inheritance model of genetics. So that's what we're doing this episode 4. A couple quick notes before we get into it today. One is that my schedule from here might get a little spotty. The times have moved a little bit, but we've been doing these on Thursday nights. I have been on family leave for a bit because my wife and I recently had a baby, but the week after next, I will be back at work, and I work late on Thursday nights. So this will no longer, after next week, it's kind of iffy, and then after that, certainly, this will not be a Thursday night thing anymore. I don't know when it's going to be, and maybe I'll just bump it to Wednesday night. That'll probably work fine, but I don't know for sure, so I don't know what the schedule is going to look like going forward. It might be try out a couple days and see what happens. So just be aware of that. The second thing is that there are some figures in here that are from a different textbook from the one I recommended before. And I actually used some figures from this book last week as well. It's the Campbell Biology textbook, specifically the 10th edition, which is old at this point. You can find it dirt cheap / free on the internet if you Google it. I think it's a really good kind of general biology textbook. It's designed for like an intro bio class. That's what I have in the past used it for. I'm actually not using it anymore, switching over to the OpenStax, the open source bio textbook, the OpenStax Biology 2nd edition. But just be aware that for some of these slides in the bottom right corner, it'll say like figure 14.2 or something. It's from the Campbell Biology 10th edition book. So if you want what I think is a pretty nice general biology textbook, I recommend that one. And eventually in the slides I'm going to be using here, it'll turn over into the figures for the new book that I'm using. I just haven't gone through and revised this stuff yet. So that's why it's using the older stuff. So those couple of quick notes out of the way, let's get into it for tonight. So as always, let's start with our outcomes for tonight. There are 3 things, or really kind of 4 things, I want you to be able to do with what we talk about tonight. First, we're going to do Mendel's model, which has 4 major components. And I want you to be able to compare and contrast those components. So basically be able to tell me, for example, what the difference is between alleles and ploidy and dominance, and be able to, know the ins and outs and be able to explain what we're talking about. Then we're going to, in kind of the second half here, we're going to talk about genetic crosses. And that means we're going to do Punnett squares. So the first thing is just be able to sequence the process of doing like an experimental cross. What did Mendel do, and how does that process work? And then specifically for making predictions and testing them, be able to use a Punnett square. That's what I mean by apply, is basically apply the technique. So be able to use a Punnett square. And if you're given a Punnett square, if you see a Punnett square, be able to evaluate it, be able to tell me what's going on and what it shows. Okay, so those are our goals for tonight. So let's start with Mendel's model. Now remember, last time we talked about the blending model. Is raw mat here? Get them in here. We'll actually next week is more relevant for raw mat stuff. If you don't know what I'm talking about, don't worry about it, everybody. So recall last time we talked about the blending hypothesis and why that's wrong. Today we're going to look at the theory, the alternative model that Mendel developed. That is the theory of particulate inheritance. And this is where the characters are determined by what Mendel called heritable factors. That's his word, heritable factors. And Mendel correctly deduced that each character in an organism, at least for the organisms he was looking at, which coincidentally happen to be the same for like us, each character is controlled by two factors, one from each parent. So you get one from mom, one from dad, and whatever combination you have, that's going to determine whatever trait you have for the traits that Mendel looked at. Now you notice there's a word missing here, and that word is gene. The word gene was not coined until 1909. So Mendel, operating in the 1860s, didn't have or invent the word gene. That comes later. He used the term heritable factors, but it means the same thing. So throughout, from here forward, I'm just going to say gene. That's what we mean, even though that's not the word that Mendel would have used. Okay, so just be aware of that. We're going to use the modern terms as much as possible, but Mendel did not have that vocabulary. So just everyone be aware of that. So now we're going to go through the 4 components of Mendel's model. The first component is that there are things called alleles, and alleles just means different versions of a gene. Now, this kind of figure, this representation is something you're going to see a bunch going forward. So let me just take a minute and explain what we're looking at here, because we didn't talk about like the cell cycle, mitosis, or meiosis, which is the process of going from a diploid cell to haploid gametes. We haven't talked about those processes, and I don't plan to. But the idea here is your chromosomes have to separate out from the parent cell into the gametes. And when they do that, they form the structure called a tetrad. And it's called a tetrad, tetra as in 4, like Tetris, they're all 4 blocks. So you have 4 chromatids, 1 chromosome, right? So this red thing, that's a chromosome made of 2 sister chromatids. Those sister chromatids are copies of each other. It's just use one as a template to make the other one. And so like that would be from mom. And then over here, you've got a duplicated chromosome with 2 chromatids from dad. And these physically connect to each other during meiosis when you're making gametes. And we call that structure of the 2 chromosomes together, we call that a tetrad because it has 4 chromatids, 1, 2, 3, 4. So that's what this picture, what this figure is representing, just so we're all on the same page there. Now, going back to the word allele, this just means alternative version of a gene, a variant at a site in the genome. So here, you typically represent these with letters when you're talking about like Mendel's stuff. It gets a little more complicated when you're doing like flies and humans, but we're just going to use letters for today. So like capital A might represent the allele for black hair, and lowercase a might represent the allele for red hair. And I recognize that hair color is more complicated than that, but just simplifying, just go with it. It's just variants of a gene. That's what we mean by allele. So Mendel said there are variants for the genes that determine what traits are present. We also have 2 factors for each character, 2 heritable factors. Basically, within each individual, you have 2 genes for each trait, or 2 genes at each locus, depending on how you want to say it. In other words, Mendel came up with the idea that we are diploid. We have 1 complete set of genes from mom, 1 complete set of genes from dad, right? So we have 2 of everything. And the notation for that is to say 2N, that's diploid. N, just 1 copy of everything that's haploid. 4N would be tetraploid, that kind of thing. So we're diploid. We have 1 copy of each gene from each parent. Now, those copies can be the same, and that's called being homozygous, or they can be different, and that's called heterozygous. So in this example right here, if hypothetically mom's chromosome, you got capital A, and dad's chromosome, if hypothetically you had capital A, then you'd be homozygous for gene A. As it is, you can see mom has given you capital A and dad has given you lowercase a, so that individual at that locus is heterozygous. Now, these different genes, right, the one from mom and the one from dad, they're located on chromosomes called homologous chromosomes. So the 2 chromosomes that are together in a tetrad, and I recognize that the terminology, there's like kind of layers and the words are all the same. So again, this thing right here, that red thing, that X, that is a chromosome with 2 chromatids. This blue thing is a chromosome with 2 chromatids. Okay, so these chromosomes, one from mom, one from dad, that have the same genes, but not necessarily the same alleles, those are called homologous chromosomes, or sometimes just homologs. So for example, in the human genome, right, you've got chromosome 1 from mom, chromosome 1 from dad, chromosome 2 from mom, chromosome 2 from dad, all the way down the line. Those are homologous pairs of chromosomes. So this is a homologous pair. The third thing that Mendel came up with is the idea of dominance. Now, this is not the only way that alleles can interact with each other. There's other inheritance patterns that we're not going to worry about today. But just for today, in Mendel's model, when you have two alleles that are different, they can be dominant or recessive. And all that means is the dominant allele is always going to determine the phenotype. Remember that word from last time? So the dominant allele is going to determine what like the physical trait is. Is the plant going to be tall or is it going to be short? Well, the dominant allele is tall. The allele that leads a plant to be short, that is recessive. So what happens is the phenotype is going to be based not on one allele from one parent, it's based on the combination. So for example, let's look at flower color. Purple, which we're going to show with a capital P, that's typically how you show dominance as the capital letter. Purple is dominant over white, which we're going to say lowercase p, right? So you could have a flower that's homozygous dominant, right? It's got two dominant alleles, so that's going to be a purple flower. You could have a flower that is homozygous recessive, two recessive alleles, that's going to be a white flower. When you have a flower that is heterozygous, it's got one dominant allele, one recessive allele. The dominant allele is going to determine the phenotype, and the recessive allele is going to be covered up by the presence of that dominant allele. So this heterozygous flower for flower color is going to be purple. It's going to have the dominant phenotype. So if you're looking at a purple flower, you don't know what its genotype is. You don't know what alleles are present. You know there is at least one dominant allele, but you don't know if it's homozygous or heterozygous. And by the way, when I do this in class, like for real, like in non-COVID times, when I'm in like a lecture hall with people, this is where I'll specifically say like, it'd be like PP, and then people start to laugh and I'll say like, yeah, I said PP, everyone laugh, and then everyone laughs and it's funny because, you know, I'm old and I make bad jokes. All right, so that's the third thing, dominance. And now we get to the really important ones, the ones that are critical for how these things go from generation to generation. They are Mendel's two principles of heredity, which are sometimes called Mendel's laws. So the two things that we call Mendel's laws are the law of segregation and the law of independent assortment. If you've had any kind of bio class that touched on genetics, you have probably heard this stuff before. Yeah, I'm hilarious. I am hilarious. There's a lot of dad jokes. I spent years practicing dad jokes before I was a dad, and now I can like actually make dad jokes, so it works. All right, so let's get into these principles of heredity because we're gonna illustrate these. They're really cool and they're really important for stuff that we're going to be doing down the road. So the first thing is the law of segregation. This just means that in your parent diploid cells, you have two alleles for whatever character you're talking about, right? Height, flower color, whatever it is. You have one from mom and one from dad. Well, those are going to separate when you're making gametes. So each gamete only gets one. So here you could have a couple of tetrads. So like here's a tetrad, a pair of homologous chromosomes. Here's a different tetrad, a different pair of homologous chromosomes. Now this just shows the process, part of the process of meiosis. Again, we didn't go through that. We didn't talk about the steps or the stages. Doesn't matter. All we need to see here is that this cell is going to divide along its equator when it's oriented like this. So each daughter cell is going to get one rather than both alleles for each gene that are present in the parent cell. That's called segregation. This is really important because this is where you go from diploid, having two of each thing, to haploid, having one of each thing, right? This cell right here for gene A, right? It's got one from mom, and it's got one from dad. Then you divide it in half. The daughter cell's either going to get the allele from mom or the allele from dad. It's going to have one copy of each gene in the genome. Therefore, it's going to be haploid, right? One of everything instead of two, which is diploid. Your gametes, right? Sperm and egg, those are haploid. When they fuse back together, you go back to diploid. So this process is critical for how sexual reproduction works because you need to separate those alleles. You need to go from diploid to haploid. And Mendel identified that. He called it the law of segregation. You separate your alleles from each other, and in doing so, reduce your ploidy from diploid to haploid in your gametes. Okay, so that's the law of segregation. And we can illustrate this looking at another trait, seed color. Now, Mendel's looking at pea plants. Yellow seeds were dominant over green seeds, so we're going to use capital Y and lowercase y. And your possible genotypes, right, in these, for this trait, you could be homozygous dominant, you could be homozygous recessive, or you could be heterozygous. But what are your possible gametes? The thing that I want you to get here is that in the adult cells, in the somatic, non-reproducing, whatever, non-germline cells, they're diploids. So you get two of everything, right? You get two of each gene. But in the gametes, you go through segregation, you separate those from each other. So now you're only going to get one. So you can't be homozygous dominant, heterozygous, homozygous recessive. You can't be those genotypes in the gametes. You can either be just dominant or recessive, and that's it. You can't have two. That's a problem. You only get one. So that's illustrating what's going on with segregation. The second law is the law of independent assortment. Now, this is a cool one because it's not exactly correct. The way that I'm going to state it is the correct way. But I want to make clear before I go into this that I'm using words and concepts in this description that Mendel didn't have. So Mendel didn't know anything about chromosomes. He didn't know anything about the concept of linkage, which we're going to get into next time. The idea that genes are on the same chromosome and therefore travel together. He didn't know any of that. He figured that each gene, each heritable factor, was on its own, acting independently of every other heritable factor in the genome. Okay, so Mendel didn't know anything about chromosomes or linkage or none of that. So he was wrong in that independent assortment is universal in the genome. But what he got right is that genes on different chromosomes assort independently when you're making gametes, when you're going through the process of meiosis. And this has to do with the orientation of those tetrads, those associations of homologous chromosomes, because the orientation of those tetrads in terms of which way dad's chromosome is facing and which way mom's chromosome is facing, that orientation is random. So you can have it one way or the other way, and the probabilities are equal. So let me illustrate this very briefly here. Here you have a state where mom's chromosomes are going up, dad's chromosomes are going down. You also have the opposite. Dad's chromosomes go up and mom's go down. Or you can have a mix where, one of mom's and one of dad's goes one way and then the opposite and each pair goes the other way. So you could have it, you have it like this, you have it like this, you have it like that, you have it like that. It's random. All of those states are equally likely. So you mix and match the combinations of alleles on different chromosomes because those alleles that are separate from each other on physically different pieces of DNA, they assort independently. The way they end up traveling is random and it's unaffected by what the other chromosomes are doing. That's called independent assortment. And again, we're talking about this in the correct way, like talking about it in terms of chromosomes. Mendel didn't know about chromosomes. He thought everything just independently assorted from everything else because the traits he happened to look at were all on different chromosomes from each other. But that was just by chance. That doesn't have to be the case. And we'll see next time a situation where that wasn't the case, which is what led people to realize that chromosomes are the things that carry genes. But now I'm getting ahead of us, so back to Mendel. How's everybody doing so far? Everybody hanging in there? Everybody following? I know I just kind of like assumed some background level of knowledge about like the cell cycle and meiosis. I hope that's okay. If that is not okay, then I want to refer you again to any general biology textbook. The Campbell Biology 10th edition has some really nice pictures, but the OpenStax 2nd edition is what they're up to now, is completely free. You can just go on the OpenStax website and download the PDF, and it's free. And that's a really nice textbook too. And that'll get you caught up if you're unsure about any of this stuff like mitosis or meiosis. So I just want to make clear the effects of these processes. So independent assortment plus another process of shuffling called crossing over, which we didn't talk about, but that's where within the tetrad you have bits of the chromosomes that can swap with each other. That leads to just new combinations of alleles. So in this simple representation, each of these bars represents a chromosome, and each pair represents a homologous pair. So we've got 4 homologous pairs here, right? And what you get via independent assortment, you've got dad's set on one side, mom's set on the other side. Via independent assortment, you're going to mix and match the chromosomes as units, right? So some of dad's and some of mom's go in each direction, right? So you got like the alleles here are now going to the same place as the alleles here, where previously those sets of alleles were separate from each other. And then with crossing over, you have little bits that flip back and forth within the chromosome. So you end up with a genome that looks like this, where you've got a bunch of crossover events that are mixing and matching the alleles within the genome. These two processes are the main sources of variation that you generate through sexual reproduction. And we're going to talk about the evolution of sex and sexual selection, all that fun stuff. We'll get to it eventually. But just for now, the big benefit of sexual reproduction is that you generate more genetic variation. And the way you do that is through the recombinatory processes of independent assortment and crossing over. And this is just a simple illustration of these processes. Now, Mendel didn't know about crossing over because he didn't know anything about chromosomes. He only figured out independent assortment, but I just wanted to mention the other side of this because those two things kind of go hand in hand as far as the diversity goes. Now, big thing I want to mention before we get to the second half of this is that Mendel didn't know any of the mechanisms behind any of this. Nothing about chromosomes, genes, DNA, none of that stuff. The cell cycle, none of it. Mendel was looking at parents and offspring and the distribution of traits and figuring out patterns and then figuring out the rules governing those patterns. So he would try to predict what he was going to get, and he came up with a set of rules that allowed him to accurately predict what the offspring were going to look like based on the parents. Okay, so he wasn't dealing with like the biochemistry of any of this or the molecular genetics of any of this. He was just looking at the kind of parent to offspring to kind of grand offspring level, looking at the results and figuring out the rules based on the observations. It's actually extremely impressive. Good for Gregor. So now let's talk about genetic crosses. So the idea of a genetic cross, it's a method for predicting the genotype of offspring. And when I say genetic cross here, I mean like on paper. I don't mean like doing it like actually crossbreeding the plants and seeing what happens. The idea is that we want to be able to make predictions. So what you want to do is actually follow the behavior of alleles and then figure out what's going to happen in the next generation or the generation after that. The big thing that Mendel did here was he could quantitatively predict what was going to happen. It wasn't just about, well, like organisms that kind of look alike, if we breed them together, their kids kind of look the same. Now, he could give you like exact ratios of expected offspring phenotypes, and that's like the big deal. So here's kind of the idea. When we do a cross, what you want to do is determine what gametes are possible in each parent. First step is you have your parents and what gametes can they potentially make? Obviously, you need to know your parents' genotypes. You need to know the parent genotypes in order to do this. That's where Mendel's true breeding lines come in that are consistent, so you know what the genotypes are. Then you determine the possible genotypes of the offspring. So if you randomly mix the gametes that each parent can make, what are the potential offspring genotypes? And then what you want to do is calculate the frequency of the possible offspring genotypes. What fraction of the offspring are going to be homozygous dominant versus homozygous recessive versus heterozygous? And phenotypically, what fraction of the offspring are going to have the dominant versus the recessive phenotype? Okay, that's what we want to do. The big thing we can do with these, which is really fun, is we can actually use these crosses to illustrate Mendel's laws. So we're not going to worry about independent assortment today because that gets ahead of ourselves, where you have to kind of do the calculations for two genes. We're not going to do that. So we're just going to focus on segregation for right now, and then we'll see how the things mix. So the type of cross that we are going to use to illustrate this idea is called a monohybrid cross. Monohybrid, mono means one, hybrid means a mix. So we're looking at individuals that are heterozygous for one character. Specifically, we're looking at a cross between heterozygotes. So here's how we're going to set this up. In our P generation, right, so think back to what Mendel did. P generation, the parental generation, is true breeding. So they're homozygous dominant or homozygous recessive. And we're going to cross those individuals together. Now, follow me in what we're doing here. If you're homozygous dominant, we're going to use P color, right? We're going to go with capital Y and lowercase y. If you're homozygous dominant, you have two dominant alleles. If you're homozygous recessive, you get two recessive alleles. So what gamete can the dominant individual make? The only option is a gamete with a dominant allele. And then you look at the recessive individual, same thing. The only option for the gametes is to have a recessive allele. Either you get a little y or you get a little y. Those are your options. So if you put a dominant allele with a recessive allele, and those are your only options, all the offspring have to be what? All the offspring have to be heterozygous. So your possible F1 generation in this example are going to be heterozygous with frequency of 1 or 100%. All your F1 individuals are going to be heterozygotes. That's great. Now we can cross the heterozygotes, and that's going to be our monohybrid cross. They're heterozygous for one trait, and we're going to do that cross and see what happens. So our F1 genotype we've established is heterozygous. Y, lowercase y, 100% every individual is the same. Our possible gametes from this? Well, every individual can now make two different types of gametes. You can make a gamete that has the dominant allele, or you can make a gamete that has the recessive allele. You can do either way. So it's capital Y or lowercase y. So now what we want to figure out are the offspring and their frequencies. Now you could mix and match these two gametes in any combination, right? So you could have a capital Y with capital Y, lowercase with lowercase, or you could have a mix. So you can have homozygous dominant, heterozygous, or homozygous recessive offspring in the F2 generation. But that isn't what we want. We want to know the frequencies. So we're going to determine the frequencies using a Punnett Square. And for everyone that's, done this in high school or college, hooray, Punnett Squares. Punnett Squares are great. All right, so let's do a Punnett Square. So Punnett Square is just a tool you can use to determine the possible offspring in a cross, and this is the important part, their frequencies. So we're going to do this together. One of the reasons we were a little late starting is I was trying to track down a whiteboard and I failed. So we're going to use the pad right here. So what we're going to do is we're going to just put this away for a second. We're going to get rid of this. And we're just going to do this on paper. Now, remember, we're doing a monohybrid cross. So we have capital Y, lowercase y, crossed with capital Y, lowercase y. So let me go back to like here. Okay, so now just my face and a piece of paper. So we have capital Y, lowercase y. We can say this is mom up here. If mom is heterozygous, you have the options of generating gametes that have a dominant allele or a recessive allele. And then over here, this represents dad's potential gametes, right? Also heterozygous because everybody is heterozygous. So you could donate a dominant allele or a recessive allele. Those are your options. So let's fill in these four boxes represent our potential offspring. So if you get a dominant, let's see, here we go. If you get a dominant from mom and a dominant from dad, you're going to get two capital Ys, right? So you get a homozygous dominant individual. Over here, you get a recessive from mom and a dominant from dad. So that's going to be heterozygous. And I'm trying to be very clear about which size the Ys are. I hope it's clear to everybody. Down here, look at that. You get the same thing, but it's the opposite way. It's dominant from mom, recessive from dad. So down here, you get another heterozygous individual. And by the way, it is convention, if you're heterozygous, to draw the dominant allele first, independent of the source. So you would draw these two the same way, even though the source of the dominant allele is switched. That's just a convention when you're doing this kind of thing. And then down here, recessive, recessive. So you get a homozygous recessive individual, two small Ys. Okay, so the important thing I want you to see here is ratios. So you have a 1 to 2 to 1 ratio, homozygous dominant, heterozygous, homozygous recessive. And phenotypically, you have a 3 to 1 ratio. These three are going to be dominant because you only need one dominant allele to have the dominant phenotype. And this one down here, 1/4 or 1 in 4 of the offspring, are going to be the recessive phenotype. So that's how we can use a Punnett Square to figure out the expected offspring frequencies in terms of both genotype and phenotype for a monohybrid cross. I'm going to leave this up for just a second. Make sure everybody is good. How is everybody feeling? I want to just go over to the chat. Did everyone follow me through that? Is everybody okay? Any questions at this stage? And for those of you watching this not live, I apologize that you have to sit through me asking if everyone's good and, anybody have any questions. Just going to wait a second, see if anybody has any questions, make sure everything's clear before we wrap this up, because we are just about done, everybody. We're just about finished here. That's really the big thing for today. I'm assuming we're good. Let's keep going. So let's go back to the screen. Okay. So we did our Punnett Square. We saw a genotype ratio of 1 to 2 to 1. We saw a phenotype ratio of 3 to 1. That's 3 dominant, 1 recessive. Ta-da! Here's the cool part. Remember last time? Those are the ratios that Mendel observed in all of his crosses. The reason we get those ratios is because of the segregation of alleles during meiosis. You get big Y, little y, and then they just mix them at whatever frequencies they're present, and boom, you get your 1 to 2 to 1 and your 3 to 1 ratios. This is exactly what Mendel observed. So remember, what I said a few minutes ago is Mendel didn't know anything about chromosomes or DNA or any of that stuff. He was looking at the ratios and figuring out the rules. And what we just did was we went from the rule and showed how if you follow the rule and just do the math, you end up with the ratio that Mendel observed. Isn't that cool? I love it when it works like that. I like that a lot. So here is the big takeaway, right? We generated a 3 to 1 phenotype ratio and a 1 to 2 to 1 genotype ratio, right? Homozygous dominant, heterozygous, heterozygous, homozygous recessive. And because heterozygotes, the phenotype that you express is the dominant allele. You end up with 3 individuals, or I should say 3 out of 4 offspring, 3/4 of the offspring with that dominant phenotype, and 1 lonely quarter of the offspring with the recessive phenotype. And that's how you can use the law of segregation to illustrate Mendel's 3 to 1 ratio. So to summarize what we covered today, what we just went through were the basics of Mendel's model of particulate inheritance, which remember has 4 major components: alleles, the idea of ploidy, right, that you get 1 from mom, 1 from dad, everybody has 2. Dominance in terms of how alleles can relate to each other. You could have dominant and recessive. It is more complicated than that. Thank you in the chat. Yes, it's more complicated than that, but Mendel did not deal with any of that stuff. We'll talk about the more complicated inheritance patterns down the road. But for now, just dealing with Mendel, it's dominant versus recessive. And then finally, the rules of inheritance, Mendel's 2 laws, segregation and independent assortment. And just remember, Mendel wasn't quite right about independent assortment because Mendel thought everything assorted independently. He didn't know about chromosomes. But as long as you stipulate that the independent assortment is on a chromosome by chromosome basis, then you're good and the rule still works. And then the last thing we did kind of in the second half there was illustrate how this stuff works using Punnett squares. So that is our lesson on the Mendelian model of genetics. So what we're going to do now is, because now this takes us in the 1860s, and then everyone just kind of forgets about this stuff for like 30 years. And it's not until like right around 1900 that people start paying attention to this stuff again. But in the meantime, there's a bunch of work done on like cell biology. So we're going to pick this up next time. We are going to talk about chromosomes and how they are related to inheritance. And we're going to do this by looking at Thomas Morgan and his work with fruit flies and eye colors. And it is super duper fun. So that is everything that I have for tonight. So what questions do you all have on this particular topic? What questions do we have on this topic for this evening? Thank you all for listening, everybody. I hope everybody found that enjoyable and also clear and not too fast or too technical or complicated. So whatever questions anybody has, I'll wait for kind of the video and on YouTube to catch up. And if you have any questions, stick them in the chat and I can put them up on screen. And here's what we're going to do. We just need to, well, no, I can just stay where it is. That's fine. Yeah, fruit flies, useful for genetics, not when they're in your kitchen. Also, fun fact, in grad school, I did a rotation in a fruit fly lab, and I will never work with anything bigger than bacteria ever again as long as I live. Anything that can fly or crawl or run away, never again. Fruit flies, pain to work with. Really cool, pain to work with. Yes, the OpenStax book, highly recommended. I really like the OpenStax biology textbook. It's called Biology 2E. It's good. No, I'm sorry. You'll have to. Sorry, Dapper. You're gonna have to. Hang on. Let me put that up because that's too good. No, I'm sorry. You're gonna have to. Come to my office hours or something because that's we're not gonna go over the whole thing. All right. What are fruit flies used for in research? All kinds of genetics. So they're a really useful animal genetic system. It's really, really useful because you can put genes in fruit flies and then like illustrate a bunch of stuff. For example, the lab that I rotated in was actually doing Alzheimer's research. And what you were doing is you were looking at mutations and different genotypes in the flies and seeing what effects they had. And then if you could tweak them to change the effects. So like you have lots of behavioral phenotypes for flies, like do they move in a coordinated way? Are they kind of agile, or are they kind of not? Are they kind of clumsy? Are they fast? Are they slow? And you could even measure their memory. So you can look at these phenotypes and see, like, are there genes that they have? Because we share a lot of genes with fruit flies. We share a lot of genes, in particular genes that affect development. So you can look at fruit fly genetic systems and see, apply that to other animals. So hypothetically, would a okay, so we're going to hide that one. Okay, so from Just a Walking Fish, we have, hypothetically, would a dihybrid Punnett Square be able to accurately represent all of human skin color diversity? Not even close. I have a really cool figure for that, actually, that I maybe show in a couple weeks when we're going to talk about heritability. And skin color, and like many other traits, is what's called polygenic. So you have many loci in the genome that affect it, not just one or two. And that leads to a continuum of phenotypes instead of discrete categories. The other thing with skin color and a lot of other traits is that it doesn't, strictly speaking, exhibit dominant or recessive relationships. It's what's called additive genetic diversity, which is a totally different inheritance pattern, where basically it's the number of alleles for a trait that you have that matters, but not the specific allele. So like, for example, for height, you could have a bunch of genes that could either tell you to be tall or short. Your height is going to be in part determined not by which specific tall genes you have, but by how many tall versus short, right? That's the idea with polygenic inheritance and additive genetic diversity. It's a totally different inheritance mechanism from what Mendel figured out. See, Dapper's is being provocative again, but the fruit flies ever become non-fruit flies? Depends on how you define them. Biochemically, one of Lenski's E. coli lines is no longer E. coli because aerobic citrate metabolism, the inability to do that, is a defining characteristic of E. coli. Like, you can biochemically test bacteria, and if they aerobically metabolize citrate, then they're not E. coli. So, like, but I see what you're doing. And I should have this on the summary, I realize, while we're sitting here. There we go. Sorry, everybody. Any other questions? Seeing none, I am going to, I think we'll call it a night. So, thank you all. I hope you all enjoyed this installment of Evolution for Creationists. Keep an eye out for more. As always, we're going to try to do this once a week. If you have not yet liked this video, how many people have liked this video? I don't know, but I bet it's not all of you. So if you haven't yet liked this video, like this video. Yeah, I see 12. I don't know how many people are here right now, but like this video and subscribe and, share this stuff and everything. Do me a solid. And also, I think I said this last time, but I'm going to say it again. I don't know if anyone saw the debate the other night, but oh my goodness, everybody, if you're in the US, like, seriously, vote for Joe Biden. All right. And like, make some phone calls to like swing states, write some postcards. Let's, like, this is serious business. Let's get that done, please. Thank you, everybody. Have a lovely evening, and I will see you, I hope, next week for episode number 5, where we're going to talk about Morgan and chromosomes. It'll be super fun. Have a great night, everybody.